Underway. Eugene, you have the floor.
Hello, and thanks for joining our webinar, Antibodies and Antibody-Drug Conjugate, Higher Order Structures Revealed. My name is Eugene Ma. I'm the Chief Technology Officer at RedShiftBio. Joining me today are Karan Shah, Principal Development Associate at Immunogen, and Ioannis Papagiannopoulos, Senior Scientist at Celldex Therapeutics. In this webinar, we will be introducing a new technique called MMS, which is ideally suited for protein structural characterization. And we'll describe the first tool built on this platform called the AQS3 Pro. We will follow up with a presentation of some case studies showing how MMS can be used to characterize mAbs as well as ADCs. With that, I'll begin. Tools for biophysical characterization of proteins certainly exist today. Many of these you may be familiar with and may even have in your labs. These include CD and FTIR for structure, DSC for stability, SEC for aggregation, UV absorbance for quantitation. But industry is always demanding more, and what they're after is the ability to measure over wider concentration ranges, the ability to achieve greater sensitivity to see the onset of conformation change, extraction of better and more actionable data from each measurement. And this means not just the ability to get cleaner spectra, but also to interpret that data in meaningful ways. On top of that, there's a desire for greater simplification of the workflow, not just in measurement, but also in analysis. To that end, we developed a new instrument called the AQS3 Pro, which we introduced earlier this year at PEGS Boston. It's capable of performing 5 key measurements in a single analysis. These include aggregation, quantitation, structure, similarity, and stability. It has over 30 times improvement in sensitivity relative to traditional IR methods and has the widest concentration range of any structural analysis platform, from 0.1 mg per mil to over 200 mg per mil. It has an integrated multi-sample capability with walk-away operation for up to a 20x savings in direct labor costs, and includes an automated protein analytics software suite, which satisfies the ease of use and detailed analysis requirements for both the operator and the scientist. The AQS3 Pro is based on a measurement technique we developed called microfluidic modulation spectroscopy, or MMS. It utilizes 3 main components: tunable QCL, which is a type of mid-infrared laser, microfluidic referencing, which takes care of our background subtraction, and advanced analytics, which converts our spectral data into structural information. The schematic on the right shows you the basic layout of an MMS system. And what you're looking at is absorbance spectroscopy. So we have a laser as our source, we have a lot of light, and so we can now use a T=MCT detector for measurement. In between is our microfluidic cell. And what you'll notice about this cell is it has 2 inputs, a channel on the left for our reference fluid and a channel on the right for our sample fluid. Much of this explanation is actually contained on our website, so I encourage you to see it. But I'll just say here that what we're basically doing is modulating the flow of fluids into our channel, and we can do this very rapidly, typically between 1 and 5 hertz. And this allows us to get differential absorbance data very, very accurately in a repeatable way. There's no human intervention involved. And so we're able to provide all the advantages of IR data, IR spectroscopy, with increased sensitivity and repeatability and the ease of use that's expected of next generation instruments. So one of our collaborators was very interested in benchmarking this instrument against their other tools. That collaborator is shown in the upper left there. It's Elion Labs, based out of Colorado. And they provide analysis services for the biopharma industry. They designed a spike test where they would introduce small percentages of BSA, which is an alpha-rich protein, into IgG1, which is a beta-rich protein. They compared MMS, which is shown in the middle column here, to FTIR, shown on the left, and CD, which is shown on the right. A 20 mg per mil protein solution was measured, The metric they used for similarity for this spike test series was weighted spectral difference, shown on the plots below. And the limit of quantitation for traditional FTIR was 22.7%. In comparison, MMS yielded a limit of quantitation of 0.76%, so 30 times better sensitivity than traditional IR methods. On the right, we have a CD which could not measure the solution at 20 mg per mil, so the measurement was taken at 1 mg per mil, where it yielded an LOQ of 3.7%. So this improved sensitivity really expands the concentration range over which we can take measurements. And for the first time, we're able to operate over the entire drug development pipeline from discovery through formulation without the need for dilution. This really simplifies the workflow. Measurements are faster, more repeatable, and there are direct labor cost savings. But most importantly, users are able to characterize the proteins as formulated at the concentrations and in the buffers of interest.
Thanks, Eugene. That was a nice overview and a great way to begin our webinar. So before we move on to Yannis's presentation, I want to remind the audience once again to submit questions for our Q&A session after the last talk. We'll try to get to as many questions as we can, so simply type your question into the Q&A box below the presentation screen and hit submit. All right, Yannis, the audience is looking forward to hear your presentation.
Greetings from Fall River. I am Yannis Papagiannopoulos of Celldex Therapeutics, and I will be presenting and discussing data and results my colleague Shannon Wren Bingham and I obtained with microfluidic modulation spectroscopy from one of the molecules we have been working with, a monoclonal antibody. First, a few words about our company. Celldex is a biopharmaceutical company developing immunotherapeutics and other targeted biologics that engage the human immune system to treat cancer and other diseases. For more information on Celldex and our company's development pipeline, which is shown here, please visit our website, celldex.com. Celldex operates at several locations in the Northeast United States. Shannon and I are at the Fall River site, at a building with a lovely view of a pond in the back, as seen here through one of the lab windows. The data I will be showing you and the analyses I will be discussing were acquired and carried out with a pre-production RedShiftBio Analytics AQS3 Pro microfluidic modulation spectroscopy instrument. Our antibody was formulated at about 150 milligrams per milliliter, actually at 148.1 mg per ml, in the formulation buffer shown. The formulation buffer composition becomes important a little later in the presentation. A dilution series was created down to 1 mg per ml using the formulation buffer as shown in the table. The differential absorbance spectra at all concentrations show excellent reproducibility. For each dilution, 2 measurements were made, but you do not see 2 traces for each of the duplicate analyses, as they are truly superimposable. The linear range of sample concentration for these analyses extends past 2 orders of magnitude, as is quite obvious when one plots the maximum of each absorbance spectrum against the corresponding concentration. The linear relationship has a coefficient of determination, R squared, of 0.999. Once the differential absorbance spectra I showed a couple of slides ago are normalized for the respective concentrations, they are essentially superimposable. In fact, so are the second derivative spectra, where minor differences could be amplified and where we see a very small difference at the lowest concentration of 1 mg per ml. Continuing with the analysis, we compare the data from the various concentrations against the data from the 5 mg per ml dilution. And the similarity is exceptional, greater than 99%, 99.2 to 99.8%, for all concentrations higher than 5 mg per ml, and actually close to 99% even for the 1 mg per ml concentration. One can get carried away with all these comparisons and forget the purpose of this analysis, which was to obtain higher order structure information. Antibodies, of course, when properly folded, have secondary and tertiary structure, and these results were generated by the RedShiftBio analytics software from the data shown previously. As you can see, the results are quite consistent across the various sample concentrations, and in fact they're also consistent with what would be expected for an antibody. I should add that this is not surprising, as we have had this same antibody formulated at lower concentrations with no discernible impact on its structure. We also obtained secondary structure information with circular dichroism spectroscopy with our JASCO J1500 CD spectrophotometer. However, in order to acquire the CD spectra, the 150 mg per ml sample had to be diluted significantly, and for the dilution PBS was used, because the antibody formulation buffer, the composition of which I showed you a few slides earlier, contains chiral molecules that interfere with CD spectrophotometry. Essentially, they swamp out the signal from the antibody. We believe that this may be the reason of the discrepancy in secondary structure calculations between CD and microfluidic modulation spectroscopy. In summary, our antibody was analyzed with a RedShiftBio Analytics microfluidic modulation spectroscopy instrument at concentrations ranging from about 150 mg per ml down to 1 mg per ml. The differential absorbance spectra of replicates for each concentration are very closely matched, so the reproducibility is quite excellent, as is the accuracy. The absolute absorbance spectra overlay very well, suggesting that the antibody maintains its secondary structure across the various dilutions in its formulation buffer. The measurements are quite reproducible over a wide range of concentrations, spanning more than 2 orders of magnitude, and so is the linear range of the measurements. Importantly, microfluidic modulation spectroscopy requires no dilutions of high concentration samples, which is both convenient and reduces the possibility of human error or the introduction of artifacts. Equally importantly, there are no interferences from the formulation buffer. The higher order structure information is, as mentioned, consistent across the various concentrations. I should add that all the data plots and results you saw in the preceding slides were generated with a RedShiftBio Analytics software. This software not only makes it easy to set up the instrument and run a number of samples unattended, but makes data review and analysis easy and intuitive as well. We wish to acknowledge from RedShiftBio Analytics Libo Wang for her expert technical and scientific help, and Jeff Zonderman for making us aware of this technology and for facilitating and driving this collaboration. And thank you all for listening.
Thanks, Giannis. That's some very nice antibody characterization work and a great way to showcase the benefits of the AQS3 Pro system. So before we move on to the final presentation, I want to remind the audience once again to submit questions for our Q&A session after this last talk. Again, we'll try to get to as many questions as we can, so simply type your question into the Q&A box below the presentation screen and hit submit. Karan, the audience awaits your insights.
Good morning. This is Karan Shah from Immunogen, and today I'm going to talk about using MMS as a tool for secondary structure assessment of antibody and their ADCs. I will start with a brief introduction to Immunogen's ADC technology and then discuss the basics of protein high order structure before moving on to specific case studies where we used MMS to study the ADC and antibody and the effects of thermal and pH stress on secondary structure. We also looked at some applications of MMS for formulation development and compared data directly with FTIR. And then finally, I'll conclude with the summary and acknowledgements. An antibody-drug conjugate, or an ADC, is a slightly complex molecule with three key components: the linker, the payload, and the antibody. The antibody in this case mainly works as a targeting vehicle that binds to the cancer cell via the target antigen. The payloads used in ADCs are orders of magnitude more potent than those delivered by conventional chemotherapy, and so must be securely conveyed to the cancer cell in order to minimize the systemic side effects. The third component, the linker, is the one that attaches these payloads to the antibody, and these linkers are engineered for stability in the bloodstream, but at the same time must allow controlled activation and release of the payload following uptake by the cancer cell. Established conjugation platforms include engineered cysteines and lysine-based technology, and depending on which conjugation platform is used, this linker will react either to the epsilon amino group of the lysine or the sulfhydryl of the engineered cysteine residues. This slide shows the basic mechanism of action of an ADC. Once inside the bloodstream, the antibody in the ADC binds to its target antigen on the surface of a cancer cell, resulting in the internalization of the ADC via natural processes. Once inside the cell, the attached cell-killing agent or the payload is released into the cell and ends up killing cancer cell, thus providing targeted cell-killing activity. On the figure on your right, you can see the ADC binding onto the cell, getting internalized, and then releasing the payload, which ends up, in this case, acting against the microtubules and killing the cell. Moving on to higher order structure of proteins and microfluidic modulation spectroscopy. So what is higher order structure? Proteins such as antibodies have complex three-dimensional structures derived from the amino acid sequence, which forms the primary structure, which folds to form a secondary structure or the polypeptide backbone that has the alpha helices and the beta pleated sheets as the dominant structural motifs. Elements of this secondary structure combine to form domains or subunits, which are the monomeric polypeptide units associated with tertiary structure. These monomers, in turn, combine to form dimers and multimers that form the quaternary structure of a protein. Collectively, the secondary, tertiary, and quaternary structures are referred to as the higher order structure of a protein. For any biotherapeutic, the in vivo activity and specificity depend upon the protein folding as defined by the higher order structure. In the case of an antibody, the conformation directly determines its ability to interact and bind with its target antigen. Also, in terms of safety, higher order structure variants may have undesirable pharmacological consequences like immunogenicity, which is why consistency is important to study in terms of higher order structure. These are the reasons why ICH guidelines recommend studying high-order structure in proteins in several sections related to physicochemical properties, comparability, specifications, etc. This slide shows the commonly used techniques used to study the different levels of protein high-order structure. For the purpose of this talk, we are going to focus on the secondary structure, which is commonly studied using far UV-CD or FTIR, both of which have some recognized limitations to them. For example, limited concentration ranges, requirements of high amounts of sample, incompatibility with certain buffer matrices, and more importantly, complex workflows that involve quite some hands-on time with sample prep and data acquisition. MMS provides an alternative to these two techniques while also overcoming some of those challenges. Infrared spectroscopy directly probes the hydrogen bonding in the polypeptide backbone, indicating whether the local structure is folded as an alpha helix or a beta sheet because of the characteristic pattern of the hydrogen bonding associated with each of these structures. The amide-1 band from 1590 to 1710 wave numbers that is associated with the CO stretching in the polypeptide backbone is particularly useful for studying secondary structure of proteins. And although IR has long been recognized as an extremely powerful tool for protein analysis, it has been unable to be completely exploited due to the current state of the technology like FTIR that has some of the challenges associated with complex workflows, reproducibility, and large amounts of sample requirements. MMS is a relatively new technique that makes it easier to exploit the inherent advantages of IR spectroscopy for measurements of secondary structure. At the heart of an MMS system is a modulating microfluidic flow cell that continuously switches between the sample and the reference streams. Measuring both the streams generates spectra for the reference matrix or the buffer that can be used for automatic real-time background correction of the sample spectra. As a result, measurements are much cleaner and are less impacted by noise and far less sensitive to background drift. Also, the analytical workflow that is associated with MMS is quite simple compared to conventional FTIR, as it requires minimal sample prep and can be automated for unmanned operation, very much like an HPLC. Now let's move on to some case studies where we used MMS to study antibody and ADCs and the effects of pH and thermal stress on the secondary structure of these molecules. To begin with, we used MMS to look at the secondary structure of an antibody and its corresponding lysine-linked ADC. In the top left figure, you have the absolute absorbance spectra is obtained by MMS for an antibody and its corresponding ADC. As you may notice from the nature of the overlay, there was excellent reproducibility between the replicates of the data for the antibody and the ADC. On the bottom figure, you see the second derivative of the absolute spectra, which increases the resolution, and now you start to see the characteristic bands associated with the different elements of secondary structure. As is expected with an IgG molecule, a majority of the structure exists as beta-pleated sheets and there's hardly any alpha helix. Some of these major bands are corresponding to beta structures. Also, there's some amount of unordered structure as pointed by this characteristic band at 1670. On the right, we have the fractional contributions to the secondary structure that are determined by the software from these different elements. The top figure is the antibody and the bottom figure is the ADC. And as we can notice, the fractional contributions for each of these different motifs for the secondary structure are very similar and comparable between the antibody and the ADC. Looking at this data overall, we were able to conclude that the antibody secondary structure largely remains unaltered on conjugation. Next, we used MMS to look at another pair of an antibody and its corresponding ADC. Although in this case the ADC was made using a site-specific cysteine conjugated chemistry. As you can see from the left figures, the absolute absorbance spectra and the second derivative between the antibody and the ADC are in pretty good agreement and good overlay. On doing further data analysis from the characteristic bands of the second derivative, we are able to tease apart the fractional contributions of the different structures for secondary structure. As can be observed, a large component of the secondary structure is made of beta sheets and turns and some amount of unordered structure with very less alpha helix. The blue and the green data points are for the antibody replicates, while as the red and the pink correspond to the ADC. As we can see, there is great agreement between the fractional contributions for each of these elements between the antibody and the ADC. Considering that the conjugation process involves a global reduction and a reoxidation step, it was good to see that the process does not actually significantly alter the antibody secondary structure even upon conjugation. Next, we wanted to study the effects of thermal stress on the ADC secondary structure. In this case, the control ADC was stressed at 50 degrees for 10 days and then analyzed by MMS. As you can see from the absolute absorbance spectra and the second derivative curves, there was pretty good agreement between the control and the stressed ADC. Although from the SEC it was evident that 20 to 25% of the ADC had already aggregated, but that did not reflect in the higher order structure as studied by the fractional contributions of the different elements towards the secondary structure. As can be seen between the control and the stressed ADC, the level of the beta sheet, the beta turns, the alpha helix, and the unordered structures were pretty comparable. Also, the similarity score from an area of overlap calculated by the software was greater than 99% between the control and the stressed material. A hypothesis for this result is that the applied temperature of 50 degrees, though sufficiently high to impact the quaternary structure and the aggregation state of the ADC, does not change the more fundamental secondary structure of the molecule, which is why there is no difference reflected in the higher order structure. So to further challenge the stability of the ADC, this time we used a stress condition of 70 degrees at 20 minutes on the ADC. As we can see from the absolute absorbance spectra, that was enough to cause some change in the secondary structure. Going to the second derivative, we were able to see that the bands corresponding to parallel beta sheets had gone down in intensity, whereas the characteristic band for the anti-parallel beta sheet at around 1620 wave number had significantly gone up in the stressed sample. Also looking at the delta analysis from the software, the area of overlap similarity score between the control and the stress sample in this case was only 76%. Again telling us that there was significant change in secondary structure on applying the stress conditions. Looking at the fractional contributions of the different components between the control and the stress sample, we can see that the anti-parallel beta sheet increases significantly as the stress sample, with a corresponding drop in the parallel beta sheet. This is a characteristic observation that can be associated with aggregate formation in a sample. Also, we see that the unordered structure is slightly increased in the stress sample. We were able to confirm this aggregation using SEC, which showed us that greater than 80 to 90% of the sample was actually aggregated at 70 degrees when stressed for 20 minutes. After looking at thermal stress, next we looked at the effects of pH stress on the secondary structure of ADCs. In this case, the ADC was stressed at a low pH of 3 for 3 days. As we can see from the absolute absorbance spectra obtained using MMS, the secondary structure dramatically changes between the control and the stressed ADC. This can also be observed from the second derivative, wherein the characteristic bands associated with the parallel beta sheets are decreasing in intensity with an increasing signal for the antiparallel beta sheets. Also, the delta analysis is able to tell us that the area of overlap similarity score between the control and the pH stressed is only 78%. Looking at the fractional contributions of the different secondary structure components, we can see that there is only a small change between the beta sheets of the control and the pH stress sample. But further resolving these beta sheets into parallel and antiparallel beta using the software shows us the dramatic increase in the antiparallel and the decrease in parallel beta sheets associated with aggregation. Again, we are able to confirm the aggregated state of the ADC in the pH stress sample using SEC, where we can see almost 75% of the ADC is aggregated under those conditions. This slide shows how we used MMS as a tool for formulation development. In this case study, we wanted to check the feasibility of an alternative formulation on an already ongoing clinical program. The new formulation provided better management of some product attributes, but had a pH that was 2 units lower than the current formulation. Now, as we saw in the previous examples, that pH can dramatically alter the protein high order structure, specifically the secondary structure. We were not sure if this was a viable option for this product. So, the corresponding ADC and the other ADC that was buffer exchanged into the low pH formulation were analyzed using MMS, and as we can see from the absolute and the second derivative spectra, they were exactly almost overlapping with each other, which indicated good agreement between the secondary structures between the two samples. This was also reflected in the fractional contributions of the different components of the higher order structure, with the antiparallel and the parallel beta sheets being very comparable between the control sample and the ADC in the low pH formulation. So in this case, the data generated from MMS was very useful in answering the question whether an alternative formulation was feasible for the current program. Although in this case just one formulation condition was looked at for screening, considering that MMS does have an automated workflow capacity, we could see how this could be easily used for early on formulation development and screening studies, where there are a large number of screening candidates and conditions that a formulation scientists need to look at for effective formulation development. Finally, we compared the data generated by MMS to a more conventional FTIR. As we can see from the figures on the left, the top figure is the second derivative of the absolute spectra from an ATR FTIR for an antibody and its corresponding ADC. The figure on the bottom is the second derivative from an MMS for the same antibody and the ADC. Looking closely at the amide-1 band region from 1590 to 1700 wave numbers, we see that the overall shape and the bands in this region correspond very well between FTIR and MMS. Also looking at the fractional contributions in the antibody and the ADC of the different secondary structure components, there is great agreement between the major components of the alpha helix and the beta structures. Although there were some differences that were observed in the minor structural elements like the turns and the coils, but this could be attributed to the differences in the softwares that were used to generate these numbers between the two different techniques. Also, the methods for curve fitting and the parameters used for peak assignments between these two were not completely harmonized that could have resulted in the slight differences in the minor components. Although the overall absolute and the second derivative spectra looked very comparable between the two techniques. So in summary, studying the higher-order structure of proteins helps to understand the folding configurations that are important in its biological function. Also, MMS provides a reliable, sensitive, and higher throughput alternative for secondary structure assessment of biotherapeutics, with the automated workflow option being a major advantage. We were also able to conclude that the conjugation of payloads via either of the cysteine or the lysine chemistries had minimal to no impact on the antibody's secondary structure as studied by MMS and FTIR. And also, the ADC secondary structure remained mostly unaltered under mild to moderate thermal and pH stress conditions, and extreme conditions of 70-degree temperature stress and a low pH of 3 were actually needed to disrupt the secondary structure of the ADCs. In acknowledgements, I would really like to thank Mike and Alex here at Immunogen for their constant support and guidance throughout all this work, and also Hardeep, Carly, and Zeke for providing all the fun samples. I would also like to thank the whole team at Redshift, especially Jeff and Lebo, who were always present to answer our questions about running the instrument or data analysis and were really helpful throughout the whole process. Thank you.
Thanks, Karan. That was an excellent presentation to end a great webinar. And I think it solidifies the value of MMS technology in today's fast-paced drug discovery endeavors.